Hello everyone, in this video I'm going to be talking about structural adaptations of a leaf. Now, the reason I'm talking about this is because at both key stage 3, 4 and 5 you do about photosynthesis. It's very hard to talk about photosynthesis and equally the topic on diffusion without talking about the leaf, the cross-sectional structure of it, and why the parts do what they do, how they're adapted to carry out their particular functions. So we're going to look at some structural adaptations of the leaf, and this is aimed primarily at key stage 3 and 4 because that's where it comes up perhaps the most. So we're going to draw a cross section of a leaf bit by bit, and I'm going to label it and explain what those parts actually do. So if you look at the majority of plants that you see outside, you can see this shiny sort of green layer on the surface. So we're going to put that into place. So we're going to start with the surface of our leaf at the top. So this is all going to be based on that leaf at the top. If we took a cross section, what you see is this sort of shiny region on the top of the leaf. So we're going to build up our picture from here. I'm going to have it going down the way. So this top layer that I'm just drawn, this is what we call the waxy cuticle. Now that waxy cuticle in evergreens is the upper surface. And what you have is basically a layer of wax. It's actually made of lipid or fat, and it gives it that shiny appearance. So the top part of the leaf is covered in this layer of wax, and the reason why you have that is to ultimately act as a waterproofing layer. It helps to reduce water loss through stomata. So in a topic that you also do at Key Stage 4 called transpiration, which is the loss of the movement of water through the plant and its subsequent evaporation, to try and reduce the effect of transpiration, you try and minimise water loss. And this is one way that the leaf does it, by having this waxy layer on the top. Now underneath that waxy layer, what you have, just draw them in place, and I'll colour them in different colours just for more of a visual. You have at the top, just beneath that waxy cuticle, a single layer of cells. It's a single layer of transparent cells, which ultimately their job is to enable light to enter the leaf. So photosynthesis requires light energy. So light's going to hit the leaf, the top surface, and it's going to go through and penetrate through these particular cells. Now these cells form what's called an upper epidermis. So those single layer of transparent cells forms what's called our upper epidermis. Now, beneath that layer, we have, now in lower school, when you're asked to draw the typical plant cell, what you've really been drawing is, in fact, this structure that I'm drawing now, and it's called the palisade cell. So you'll notice I've drawn here very long column-like cells, and these little green dots that I'm just putting in place around the outside, these are meant to represent what are called chloroplasts that contain chlorophyll, a pigment designed to trap the sunlight. Now here's a little bit of extra info for Key Stage 4. You are also, as well as having chlorophyll, there's something called accessory pigments. Examples include carotenoids and xanthophylls. So, and these accessory pigments allow for the absorption of all parts of the light, because the leaf cells don't just use well, leaves appear green because they're absorbing all parts of the light except the green, but there are accessory pigments that can pick that up. So it's designed to maximise the amount of light that the leaf takes in. So from all the way from the red all the way to the violet spectrum. So what we've drawn here is a layer of palisade cells. These are long and thin cells packed with chloroplasts that contain chlorophyll. And their job is to provide, ultimately, a large surface area for the absorption of light. And by having them densely packed in, it sort of creates a short diffusion distance for gases. So these are what we call palisade cells. The information I was just saying about the accessory pigments only really appears in key stage 4, less so at key stage 3. It's just making the point that this chlorophyll pigment does trap light, but there are other pigments that do exist. Chlorophyll isn't the only one. So we've put our waxy cuticle in place, our upper epidermis, and beneath that are palisade cells that will trap the sunlight. And what you find just beneath those are these cells. I'll just draw in a few, and again I'll put some chloroplasts in them. These are what are known. I'll just put these black dots that I've been drawing are the nucleus, or the nuclei rather, of each cell. Control them all. What you have is a layer of spongy mesophyll cells. Now these cells are irregular in shape, so clearly it's not the best drawing, but we can see that they're all different shapes, and there's lots of air spaces between them. So where I'm just colouring in sort of blue, these are air spaces, so gaps, if you like, between these spongy mesophyll cells. And having those air spaces speeds up the diffusion of gases into the palisade. So in photosynthesis, the plant needs CO2, so carbon dioxide comes in, it goes through those air spaces and into here, the palisade cells. Post photosynthesis, when it's made oxygen, the oxygen is released, so it comes from the palisade, can go through those air spaces and out the leaf. So we're going to talk about now how those gases actually get in and out. So we have this layer at the bottom. Now, it's very much like the upper epidermis. And as you might imagine, these cells here, I'm just colouring in, form what's called our lower epidermis. Now you notice I've left some gaps there. I've left some clear gaps, and it's because of something very special about these particular gaps, if you like. So you have holes on the underside of the leaf. They're called stomata. So these holes here, I'll label this in red. They're called stomata. Stoma, S-T-O-M-A, is one, and stomata is plural. And those holes are surrounded by, so these particular cells that I'm just labelling, in fact I'll colour them in a slightly different colour. I'll colour them in purple. These cells I'm colouring in purple, they're called guard cells, G-U-A-R-D, guard cells. And they are able to allow, or to open up essentially, to create this hole. And this, through this hole, through this stoma, which gases can simply diffuse in or diffuse out. And clearly for a leaf, it's kind of got to weigh up how often or frequently it's going to open those stomata, because you can lose water through them. So we have what's called our lower epidermis. It is a layer with lots of tiny pores or holes, called stomata, that are open and closed by guard cells, and the job of that is to regulate the entry of CO2 and the loss of water from the leaf. I should just say more generally, because I didn't really start with this, but the leaf as a whole is very thin and very flat. And in another video I've talked about diffusion, and that for diffusion you kind of want a large surface area, large concentration gradient, small diffusion distance. That's why this leaf has a very, very thin cross section, because it has a big surface area for the absorption of light. That helps because it's very wide, and has a very short diffusion distance for the gases, because it's so very thin. Okay, there's one final thing I really should just add in this whole diagram, and I've not included it yet, but I'll put it in just now. And I'll just draw one example. In fact, I'll draw it just over here. I do have another video more specifically about this structure. So within a cross-sectional area of the leaf, you'll see what's called a vascular bundle. And it's made up of vessels, we call, and I'll label this at the top, xylem and phloem, which together or collectively we call veins. Now they are made up of, dead hollow tubes, they're the xylem, and living cells called phloem, which are continuous with the stem and the root. And their job, for the xylem, the job of the xylem is to transport water up and down the plant, and the phloem, I say up and down, for essentially from the root towards the leaf, and the job of the phloem is for the transport of minerals and sugars within the plant in a process called translocation. So you have, within this cross-sectional area of a leaf, this vascular bundle. Vascular mean similar to like our veins that we look at in our own bodies. This vascular bundle composing of xylem at the top, that's the blue portion, and phloem at the bottom, that's the sort of the pink coloured section. And the job of those xylem and phloem are to enable the transport of water and minerals and sugars in the plant. I just want to finish by showing one particular slide, and it's this, and this is actually what you can view with an electron microscope. This is the underside of a leaf. Now it's very simple. In a classroom setting, using a piece of sellotape and nail varnish, you can actually put nail varnish on the underside of a leaf, let it dry, put a piece of sellotape over it, peel it off, and you can take with it an actual layer of cells from the bottom of this leaf. And that's what we've done to get this picture. And with an electron microscope, you can clearly see we've got these guard cells at the top, and this stoma, this hole right in the middle of them, which will open and close. So I just thought it's interesting just to see what it actually looks like in real life. So there is a cross-sectional structure of the leaf. We've talked about the adaptations of each part and the job that they play. Okay, I hope all that helps.